Hi, everyone. I'm Ruby. I'm doing my PhD at Maquarie University and also at the Royal Botanic Gardens in Sydney. My supervisors for my PhD are Rachel Gallagher, who's now at Western Sydney University, and Hervé Sakay, who's at the Botanic Gardens. And so I'm going to be talking to you today about flowers, floral traits, and Flora Connections. So the first two really relate to my PhD, which is all about flowers and floral traits. And then the third is Flora Connections is a project I've been working on as a research assistant, which is a citizen science project. And I'll explain a bit more about that later. So first up, to start, I just wanted to add my own acknowledgement of country. I live and work on the unceded lands of the Wallumattagal people of the Darug Nation. So it's people of the river, of the Parramatta River. So yeah, I just want to acknowledge that these lands weren't ceded and pay my respects to elders past and present. And a bit about me quickly as well. So I'm in my second year of my PhD. And I'm also working as a research assistant for the Flora Connections project. And I also, before my PhD, was working as full-time as an ecological consultant. And I'm still doing that a little bit in my spare time. So I wear a lot of hats, basically. But sort of overall, I'm basically a plant ecologist with a keen interest in bees, but pretty much focused on the plants for now as well. So that's kind of where I'm coming from. So today, like I said, I'm going to talk about flowers, floral traits, and the Flora Connections project. So first up, I'll talk about the ecology and the evolution of flowers. And I'll zoom in on the first chapter of my PhD, which is really about flowering periods in Australia, as a kind of example of more of the macroecology of floral traits. And then at the end of my talk, I'll talk a little bit about the Flora Connection Citizen Science Project, which is still ongoing, but we're getting close, and hopefully soon I'll be able to send out invitations for people to get involved. So flowers, basically. I don't know if you recognize any of these. This is just a small subset of the enormous diversity of flowers, which kind of vary in color and shape and scent and kind of floral strategy as well in terms of pollination in every imaginable way. These flowers kind of go on the left up here. These are some of the most basal flowers in terms of evolution, some of the flowers that diverged earliest. And it kind of goes through some of the major groups. This isn't nearly all of them, of course. There are so many. And this variety, hang on, I'm just going to change my little thing because it's getting in the way. Okay, sorry. Yeah, this variety really comes back to both ecological and evolutionary forces. So how did flowers get so diverse? It's kind of, there's a whole lot of different things at play in this, and obviously pollinators is one of them, but also a whole lot of abiotic factors. And so I'm going to take a little bit of a deep dive into that next. Kind of start with the flowering plants. So flowers are the key feature of the flowering plants, also known as the angiosperms. This is kind of a fairly simplified phylogeny of plant evolution. So start off with sort of hornworts, the liverworts, and the mosses, which collectively are known as the bryophytes, and they diverged first in plant evolution. And then after that, so none of these have flowers, none of them have even seeds. They have their own quite unique ways of reproducing and unique life cycles. After that, we have the peridophytes, which are the lycophytes and the ferns. So they reproduce by spores. Again, no flowers, no seeds. And all of these life stages are quite dependent on water for the sexual reproduction. I think in hornworts or liverworts, often you need a little drop of water to move a spore across. and in the ferns as well. I think they're quite dependent on water for moving those spores and affecting sexual reproduction. So it's kind of a less efficient way of reproducing. Then we get into the seed plants. So both gymnosperms and angiosperms have true seeds, but only the angiosperms have flowers. So the gymnosperms, a lot of them are wind pollinated, and they do have different structures for sexual reproduction, and some of them are in fact insect pollinated. There are a lot of cycads, for example, that are insect pollinated. But you only really see the true sort of serious adaptation to different forms of animal pollination in the angiosperms, which are kind of down here at the bottom of this long phylogeny in terms of a group. But when you look at the number of species, they make up about 90% of land plants. So chances are, if you're looking at a plant, and you have no idea what it is, there's a 90% chance that it's an angiosperm or a flowering plant. There's about 300,000 known species in about 435 families. All of these numbers tend to change as taxonomists sort of hone down and whittle our knowledge of all the plant clades down. And this is just a simplified example of sort of the basal angiosperms. So again, these are the ones that kind of branched off first in terms of evolution. And then the core angiosperms are the monocots and the eudicots, and these numbers are the number of species in each group, I think. So that's where the huge diversity is, really. And these are all kind of considered more basal. So the really defining unit for the flowering plants is the flower, like I said. And the flower is the sexual structure of the angiosperm. So it's how they affect cross-pollination and produce seeds and produce the next generation. All flowers have this same fairly basic structure, despite there's an enormous diversity, but when you kind of pare them back, they all tend to have what we call four whorls. So the outer whorl is called the sepals, and it's also called the calyx collectively, but each individual member of the calyx is called the sepal. Then the next whorl in is the petals, which is also known as the corolla. Next one in, you have the male part of the flower, or which is the anthers, which release pollen, and also the filament. Together, they're called the andrecium. So again, it means like the male reproductive parts. And then the very center of the flower, you have the female parts. So you have the stigma and the style, which leads down to the ovary. And collectively, they're called the gynecium. So again, the female reproductive parts. And so if you look at any flower on any flowering plant, it'll have this same basic structure, although there's a lot of variation. So often parts can be missing or combined. This is an example in a real-life Australian plant, Iliocarpus holopetalus. It's a very tiny little flower, but you can see the sort of furry structure on the outside is the sepals. And then next step in, you've got the petals. Next step in, the anthers, and then right in the center, you've got the stigma. And so again, that's that four basic whorls, which make up pretty much all flowers, but which have been varied in huge and different ways to create all the variety of flowers we see today. So some of that variation has happened through the process of evolution. So this on the left here is a reconstructed flower. So it's not a real flower. It's a hypothetical flower that has been reconstructed by my supervisor, Hervé Soquet, and a whole heap of his colleagues. They took the traits of I can't remember exactly how many, hundreds at least, of the current flowers that we know today. They kind of reduced them down to their basic parts. So how are the petals arranged? How many parts do they have, or which parts of the flower are missing? And they did a thing called ancestral state reconstruction to try and infer from all of the current day flowers what the ancestral first flower might have looked like. And this is what they came up with. So it looks a little bit like a magnolia flower, or also I think a little bit like a water lily. This is a water lily up here. But it's hard to see, but it does have, it's a bisexual flower. So in the middle here, that's the stigmas. There's multiple. And then around them, you've got the andrecium, or the anthers. And then arranged around that, the colour, they couldn't work out the colour. Colour is super variable in flowers. So the colour is kind of just invented for the purposes of this illustration. But they did work out, more sort of structural things like the arrangement. So they worked out that the petals, or I think in this case they're tepals, so they're a combined, the sepal and the petal are kind of a combined unit, are arranged in this spiral formation, which creates this really big, loose sort of looking flower. But yeah, how did we get from this one ancestral flower to every single possible variation you can imagine of that in flowering flowers today? And this is just an illustration of, the sort of path that this evolution might have taken from the very early angiosperms all the way down to these super, super diverse rosids and asterids today. And one of the answers of that is through ecological processes. So evolution and ecology, I feel you can't ever really totally separate them. And so obviously the big sort of purpose of flowers is to affect reproduction, and they can do that in a whole variety of ways. They can self-pollinate, or they can cross-pollinate, and they can use bees or birds, or I think there's even slugs, or there's water pollination in some sort of underwater plants where they just release their pollen underwater. But yeah, so often in a lot of land plants, they want to attract pollinators, and that's how they end up with all these different bright colours and interesting shapes that kind of make it slightly more difficult for some pollinators, but not others, to access. That's a really common theme in the orchids. But they can also be pollinated by wind, which leads to this extreme reduction. So you can barely even see anything that looks like a petal. You just see the anthers. Similar in this one over here, even though it has a bee on it, this is a mostly wind-pollinated species. So there's no petals or anything to obviously attract pollinators. Obviously, they still come and steal some pollen every now and then because they're hungry. So flowers are, ecologically really shaped by these interactions with different pollinating agents, but they're also shaped by the sort of physical surroundings around them and abiotic factors like water availability, because it's quite expensive to produce a flower, and especially to fill it up with nectar every day. That uses both a lot of water and also a lot of the energy of the plant to produce sort of the rich sugary reward that pollinators want. And as well as all those sort of abiotic factors, there's also flower enemies, like this little beetle over here, which might look like a pollinator, but actually, I watched it for a while. It's just sitting on there and munching away at this flower and having a good old feed. And unless it might visit another flower, or it could just be there to fill its belly and not actually do any sort of useful pollination. So it kind of takes me to the central question of my PhD, which is very, very broad, but it is how and why do flowers vary, both in space and in time? And so obviously that is kind of a huge question to try to answer. But I've tried to narrow it down through my PhD by focusing on different particular flower traits or characteristics of flowers. So some of these trait is basically anything you can kind of clearly define and measure about any sort of organism. So size is a really obvious trait where you can kind of clearly define, okay, this is how we'll measure size. And then you can measure it across a heap of different species and kind of make inferences about how size evolves, what kind of influences the evolution of that trait. For my PhD, I'm also looking at traits like flowering time, the period that something flowers for, and also the longevity of individual flowers. It's kind of two different measures of time in flowering. Also things like the shape of flowers, so the symmetry, and kind of slightly more generalized traits or inferred ones about what pollinates a flower, or what syndrome that flower has, what kind of pollination strategy it's going for, whether it's trying to attract an animal or whether it's just sticking its anthers out and hoping the wind will carry the pollen away. So this is kind of a breakdown of my PhD chapters so far. Or, you know, most of this is what I'm planning to do. So I'm going to be talking mostly about my first PhD chapter today, because that is mostly finished and written and currently in review at a journal. But currently I'm working on chapter 4 down here, which is sort of all about the evolution of wind pollination across the flowering plants. And I'm also hoping to do some field work later this year to work on the relationship between floral symmetry and the floral longevity, or how long an individual flower lasts. And these two chapters 3 and 5 are kind of for later down the track for next year, hopefully. So I'll be trying to take my work on the evolution of wind pollination and extend and look at it in focus across Australia and how that varies across Australia ecologically. And then hopefully also have my final chapter, I'm hoping to really focus in and look at just flowers in eucalypts, and particularly about whether Western Australian eucalypts, which tend to have those huge flowers and the huge fruits, strongly diverge from Eastern Australian eucalypts sort of overall, which, you know, whether Eastern Australian flowers are all small or if it's actually just a bit variable. And yeah, what influences that difference in flower size. So yeah, that's just a lightning overview of my PhD. And now I'm going to focus in on my first chapter, like I said, which is about flowering periods across Australia. So this chapter I did both with my supervisors, but also with some really great collaborators. Greg Gurin is from the Terrestrial Ecosystem Research Network and the University of Adelaide. Ming-Kai Jiang is out at Western Sydney University, and Daniel Folster is at the University of New South Wales. So it was a really collaborative project. I got some really great data to work with. Whoops. It seems to be moving by itself. Okay, so flowers are, like I've explained, you know, super interesting, super variable in lots of ways. And also flowers can play a really important role in ecological communities, because they're food for all those pollinating species, but also for the floral enemies, other species, as well as being really important to plant reproduction. So the dynamics of flowering can really change an ecosystem throughout the year as different species flower at different times. This is the same site in 2 different months of the year in 2020 up in Ku-ring-gai National Park. And you can kind of see, you know, on the left in August, there's all sorts of different species in flower. But as we tick over just 1 month into September, a lot of those species have finished flowering. And so that flowering, you know, can happen really at a community scale. Whole communities, you know, kind of have all the plants in that community have different flowering periods, and that can interact to create a flowering period for the whole plant community. So I was really interested in looking at this across Australia, and looking at how flowering periods vary across all of the temperature and precipitation sort of climatic variation that we have across Australia. So we're probably pretty familiar, but we've got, if you break it down, we've got 8 broad biomes across Australia. The biggest one is the deserts and the xeric shrublands, followed by the sort of tropical savannas up in the north. And then the smallest one is the montane grasslands and shrublands of Kosciuszko National Park. That's a teeny tiny little spot down in Southeast Australia. And across all of these biomes, we have a fair amount of variability in terms of both mean annual temperatures and mean annual precipitation. The white hole here is areas where basically there is nothing to record temperature or precipitation. So that those areas have been excluded from all my analyses, unfortunately, because there's no weather stations out there, or there haven't been for long enough at least. As well as these sort of mean climatic measures, you can also look at the predictability of climate. So predictability measures how variable something is both month to month, but also year to year. And as you know, with the El Nino and La Nina sort of pattern of climate in Australia, we can have quite variable climate from year to year. So, this has got some sort of timer on it. So I looked at both the predictability of temperature, which is fairly predictable across Australia, but especially when you get towards the coast, and precipitation, which is super unpredictable, especially in inland Australia. Gets a bit more predictable sort of up in the tropical north and along the coast in the south. So I wanted to combine those sort of climatic measures and look at how that might be driving flowering periods across Australia. So to do this, I got data from the Terrestrial Ecosystem Research Network. They send out ecologists to locations kind of almost all across Australia. They're particularly focused on the rangelands, so they've got a lot of locations in inland Australia, but they're also expanding into forest plots as well. So it's called the AusPlots Initiative, and it's really fantastic source of data because they go out and they measure 100 meters by 100 meters at a location, and they very precisely record every plant species that occurs at that location, and also the cover or abundance of that plant species. They kind of go at each meter, they will determine what plant species occurs at that exact point, and they'll do that for, I think it's 10 north, or maybe it's 5 north-south transects and 5 east-west transects. So you get these fantastic measures of the plant community and the abundance of different plants in those communities. And I wanted to combine that data with data on those species' flowering periods, which I got a lot of from a database that both my supervisors are involved in, called the AusTraits database, which is a database of traits for the Australian plants, and it has heaps and heaps and heaps of both Australian species and just a lot of different trait data. It's a really fantastic source. I went through and got data out of that database for a couple of thousand species, and then I went through and error checked that data and also filled in the gaps basically for the species that didn't have them. And for those, I looked at the state floras and in a few cases, state herbarium records. And so the flowering period that I measured was kind of the months of the year in which something flowers, and it was the number of those months. So this is Rostellularia ascendens. It doesn't get down quite as far as Sydney, but it is sort of from up in Western Australia all the way across to northern New South Wales. It's got very adorable little pink flowers with a little lip. And so Rostellularia at different populations will flower for possibly a few weeks or a couple of months at a time at each of these different locations across its range. But I pulled all of those records. So I just wanted to know across its entire range in Australia, how many months of the year has it been recorded flowering in? So that means that for Western Australia, it's been recorded from January to October. But in the Northern Territory, it's been recorded flowering in every month of the year. So that gave me a final length of flowering period of 12, like every month of the year for that species. And it's kind of a very general measure, but when you're trying to look at across biomes and across things, unfortunately, I can't go out to 600 different sites and sit there and record precisely at those sites when everything is in flower. That'd be a lovely project, but I think it might take me a couple of decades. So I had a few predictions for how these flowering periods, so for each of these sites, I took these length of flowering periods and I basically calculated a mean for each site. And that mean was weighted by the abundance of that species in that plot. So if the Rostellularia was super abundant in that plot, its mean would be weighted by that abundance. It would count for more for that plot. So I had a few predictions for how the flowering periods might be responding to climate across Australia. Basically, I thought where there's a lower mean annual temperature, you'd get shorter community flowering periods. It's because in places with low mean annual temperatures, like this is Kosciuszko, there's a really short time period of the year in which species can flower and in which pollinators will be active and able to visit flowers. For opposite reasons, I thought that communities that have lower mean annual precipitation would have longer community flowering periods. And that's kind of because these communities that have the lower mean annual precipitation, they have to be really responsive. So whenever it happens to rain throughout the year, they have to be able to very quickly grow, in some cases, flower, produce seed, and complete their whole life cycle while that water is available. And for similar but opposite reasons, I thought that predictable temperatures and predictable precipitation would make for shorter community flowering periods. And that's because where the temperature and the rainfall are quite predictable, the species there can really time their flowering in line with that predictable climatic cues. And so you'd expect that overall they'd have a more concentrated period of flowering rather than this sort of very responsive year-round potential flowering. So when I looked at the averages by biomes, it kind of all made sense, which was a relief. If you look at this is the community weighted mean for the lengths of flowering periods. So 12 here means 12, that an average of all the species in the plot flowering for all months of the year, potentially. And 1 would be all the species in the plot flower for only 1 month of the year. So when you look at the deserts and xeric shrublands, they had the longest community flowering periods with a fairly long tail. And when you looked at the montane grasslands and shrublands, they had, as predicted, a much shorter community mean flowering period. So it's much more concentrated, which makes sense because they're under snow for half the year. So that was a good relief. And you look at this in a map form. I'll give you a second to digest this because it is a bit, it's a lot. These are the mean community flowering periods across Australia. You can kind of see, like in central and northern Australia, is where you get more of the longer community flowering periods. And there's a bit of a trend as well towards southern Australia that you get shorter and more concentrated flowering periods. So when you look at this by the sort of monthly pattern of flowering, it makes sense as well. You can see that this pink line here is the montane grasslands and shrublands, and that totally stops flowering in the middle of the year when it's all under snow, and has this very strong peak of flowering from December to February. Where we are in Sydney counts as temperate broadleaf and mixed forest. And so that has this sort of fairly seasonal dip in flowering in the middle of the year, and an increase kind of towards November seems to be the peak, but also December, January are fairly high. When you look at the deserts and xeric shrublands in central Australia, it's just this kind of very wavy, almost flat line with a slight peak. So those communities seem to really flower possibly at any time of the year, basically in response to rainfall when conditions just happen to be right, rather than having this much stronger, more seasonal flowering. Just quickly, this is the climate mean. So again, it all makes sense. As you get higher mean annual temperatures, you have longer community flowering periods. And you can see here that blue is the highest mean annual temperatures in Australia, up in the tropical north. And again, sort of on average, some of the longest community flowering periods are up there. And again, down the bottom is the montane grasslands and shrublands with the shortest community flowering periods and some of the lowest mean annual temperatures. Precipitation, similar but opposite. So the lower the precipitation is, the lowest is in the desert xeric shrublands, and that again has on average higher or longer community flowering periods. And again, you've kind of got some of the tropical savannas, but also the montane grasslands at the shorter end of community flowering periods, where they've got more rain, so more resources to flower when they need to. When you look at predictability, again, as predicted, the more predictable your temperature or your precipitation, generally the shorter your flowering period is going to be. So less predictable temperatures and precipitations lead to much longer, more responsive flowering periods. So in conclusion, climate really shapes the flowering strategies of plants in different communities. So when you've got predictable low temperatures and high precipitation, you can have shorter flowering periods, and you can time it to those nice, good conditions, both for pollinators, but also for the stress of flowering. And when you're in harsher conditions, you've got unpredictable climate with high temperatures and low precipitation, you'll tend to have longer community flowering periods, as everything is kind of just has to be super responsive to conditions and flower whenever it gets a chance. Some of this has implications under climate change. You'd expect that these sort of community flowering periods in response to climate change might have to shift, both by species on their own adapting flowering. So as temperatures get hot, they might just flower for longer in the year. But also more drastically by local extinctions or range shifts, by species moving across the landscape as the conditions become unsuitable for them. And so that might really see a shift from these shorter flowering periods to overall longer flowering periods, as species have to become more responsive to less ideal conditions. So that wraps up the first chapter of my PhD. And I've got a preprint published at the moment. It is in review with a journal. I've still got all the rest to go, but having a lot of fun with it. So yeah, it's good work. So I don't actually know how much time I've spent because I haven't set a timer or anything, but I was just going to talk quickly at the end now about the Flora Connections citizen science project. So Flora Connections has been funded by the Federal Department of Agriculture, Water and Environment as part of their response to the bushfires in particular. They've funded a range of citizen science projects and also bioblitzes, which are happening in a month, I think. But Flora Connections, the aim is really kind of goes beyond the bushfire response as well. And the aim is to try and connect community plant knowledge, or citizen science plant knowledge, but also really people's knowledge of the plants that they see regularly in their own area, that they know really well from their local area, and connect that plant knowledge to government, basically, and to, in particular in government, people who are making decisions about the conservation of plants. So making decisions about whether species are listed as endangered or critically endangered or vulnerable, and also making decisions about ecosystems as well. But I think our focus is particularly on plant species, because ecosystems get pretty tricky to define and survey. So the project's main aim is to try and help citizen scientists to do assessments of plants in their local area, and particularly rare or potentially threatened plants, especially those that are currently being considered by the scientific committees. The Federal Scientific Committee, which meets pretty regularly and makes a lot of those decisions based on scientific evidence about whether a plant species should be considered endangered or whether actually it shouldn't be considered threatened at all. And those decisions come from really the best data that can be found about how many populations of a plant there are and how many plants there are in that population, what's happening over time, so whether the populations are increasing or decreasing, and also particularly what threats there are to a population. So that's why I've included a little example here of a threat assessment for a plant species. The threats can be actual current threats, so things that are happening now and impacting it, or they can also be potential future threats, which climate change kind of comes up a lot in that one. I chose this slide. This is a photo of a threatened species that I think occurs in your backyard, the Grevillea parviflora, subspecies parviflora, the small flowered Grevillea. This one's from Voyager Point. So we're kind of interested in assessments of species like this Grevillea that are already listed as threatened, but also ones that might not be listed, but might actually be under threat and need more attention. And we're trying to create both a website and just a basic printable data form as well to go with that, and a whole lot of resources for how to collect that data and fill it out. And try and enable people to get that knowledge about what's happening in their backyard up to the people who are making decisions about it. Because I think there's a feeling at the levels of the scientific committees that there's a lot more known than they ever hear about. And, we kind of need a lot more connections, basically, to try and get that knowledge out there and considered. So currently, we're still developing the guide, the data sheet, and the website for Flora Connections. Hopefully it's going to be up in the next few months, and I'm going to start trialling assessments with groups. I'm going to be, I think, at the big bushfire bio blitzes. There's one happening in the Blue Mountains at the end of this month, and also one down at Murrumarang National Park on the south coast. And this project I'm just a research assistant on this project. I'm working for Rachel Gallagher, my supervisor, and also she's working with Erin Rogers, who's a fantastic citizen science person at the Atlas of Living Australia. We're really trying to make it a collaborative project. So if anyone has any ideas for it as well, I would love to hear them. We're kind of trying to work out to make it as accessible and easy to do as possible, but also to make the data as firm and usable as possible at the other end. Yes, I think that is everything. I have no idea how much time I've spent. Please tell me if you have any questions. I have not looked at the chat. And if you want to get in touch with me, it's my email, my Twitter, my website. The climate shapes flowering periods across plant communities. I have a preprint up, which is kind of a new thing for me, but it's open access, so you can download and read all about it, basically. It hasn't been peer reviewed yet, so it's still in peer review. And these are my acknowledgements as well for everyone I've worked with.
If you enjoyed this video, why not subscribe and view other great content? New videos being added all the time.